Hi, Dr. Chala.
It is, this is the second video.
Yep.
And last week we had a presentation from Dr. Yoder. We learned how to work with ZFin and Ensemble, and we had a tour of the zebrafish facility.
Yep, we're not group 4 like we said last time. We're actually group 6.
Oh, okay.
Hi.
We're group 6. Nice to know.
So I guess we should just start talking about Dr. Yoder's presentation.
Yeah.
So a lot of things he talked about were just the different cilia diseases, and it was kind of interesting to see how something that kind of isn't that talked about a lot, how it affects so many different things. Like with our gene, we're looking at the FOP gene, which has to do with anchoring in the microtubule. So when you disrupt that gene, it hopefully will see that, well, kind of not hopefully, but we might see that there's like complete dysfunction as a result of these microtubules in the cilia being out. So I think what was one of the ones that like disorders we saw? It was like a heart disease, right?
Yeah, it had to do with the gene in humans, how it altered the amount of red blood cells produced. I think it overexpressed the number of red blood cells that was supposed to be in there.
Yeah, so it was a type of cancer, I think.
Okay.
I think that's what it said.
Yeah, it had to do
Yeah.
with bone marrow.
Yeah.
Yeah, so that was pretty interesting to see. So.
Yeah, it was really interesting. I didn't know that there were like cilia on just most cells. I mean, I knew about like in the throat and places like that, but I didn't know that like the majority of cells have cilia.
Yeah, that was really interesting to look at, and hopefully when we create mutations in the zebrafish, we'll find some cool phenotypes, because it was really, I mean, at least I think it's really cool that we have the chance to, like, do mutations on these fish that aren't really looked at in zebrafish. So, like, we're going to be the first ones basically doing that, which is, like, it's kind of weird.
That's really cool, yeah.
But it's really cool, and I think, I mean, I'm really excited for that. ZFin was really helpful to kind of get insight onto our gene. I think that ZFin had a little bit less information than Ensemble.
Yeah.
Because I think Ensemble was a more like comprehensive database.
Yeah, Ensemble, it was really cool to see the exons, and it also showed the splice pairs. Right, yeah. And so that was really cool that they have chronicled all the different exons in that many different genes.
And I think just like looking at the different, like the exons and then the splice transcript, I think is what it's called, like the different variations of the gene that exist. I mean, I think it's just really cool that they have it all in one place, and you can see the complete sequence of that gene, and it's just like very useful when we're like researching stuff. And then they give the function and like what's known about it, and yeah, and I think that it's really helped us with our research. And I know that research, but I mean, I know we have a lot to learn and stuff, but hopefully within the next few weeks we can learn more about like what exon you want to look at, and yeah, because that's going to be what we're going to do for this semester.
So also we did the zebrafish facility makeup tour on Friday, and that was really interesting with Dr. Susan Farmer. She was very interested in the topic, and it helped us be interested in it too. So it was really good to see the applications for the zebrafish, because like that translational science between like the from the animal model to the human model, and how she talked about how current research is moving towards like moving very, very specific diseases that people have into a very specific kind of treatment that they could possibly have, like that one
With Dr. Alexander.
Yeah, with the one boy with muscular
Dystrophy, yeah.
Yeah.
He has a special type of muscular dystrophy, and so they're doing research specifically for his disease, which is really cool that, yeah, as you were saying, like medicine that's specific for one disease, yeah.
And I think actually, I don't know if this is right, but I think my sister said that that's actually what she's working on. Like, she's the one that's like looking at that gene mutation.
Okay, that's cool.
So yeah, I thought that was like so cool, and like that kind of cutting edge technology is stuff that us as undergrads, we have the opportunity to like kind of get our hands on and get started.
That's
which
really
is like so cool. Like,
yeah, that's awesome.
This is stuff that like I wouldn't even imagine.
Yeah,
like, would
definitely
be
in high
possible
school.
first of all, and then
Yeah.
us like coming out of high school, and now we're just like doing this cool stuff.
Yeah, definitely in high school I would not have thought that I'd be taking a class this semester where we get to mutate genes. Like,
Yeah,
that's
me neither.
really cool. So
Yeah. So something that we were kind of thrown off by was the number of animals that was in the facility. Like it was just
Yeah.
a very big surprise, and obviously since we're all science majors, we're aware of the use of animals in the laboratory. But yeah, we just didn't know how huge the animal facilities were in UAB.
And it was really interesting to see, like, when we were talking to Susan Farmer about, like, the maintenance and upkeep of the animals.
Right.
Like, I didn't realize that so much attention to detail would be, like, put into caring for them, which, like, it makes sense when you think about
Right.
it.
Yeah.
But, like, oh my, there's so many things. Like, you would have to have red light because
Mhm.
otherwise it would disturb the fish.
Yeah.
Yeah.
Even like they did experiments on which type of pebbles that they put at the bottom, what color was best for like breeding and things like that, which is just something that I wouldn't have ever thought of like doing, and
Yeah.
they've done it. So it's really interesting to see all the thought on just the smallest details go in.
Mhm.
Also, I didn't realize how quickly after fertilization, like the fish, they actually look like fish. I mean, after she showed us a petri dish of eggs that had just been fertilized that morning, and another one that was 24 hours old, and the differences were insane. It's really fascinating.
And one of the things, I had a question I asked Susan Farmer about, like, I noticed that some of the ones that were just born, like the fish that were just born, they weren't moving. And then, like, a few days afterwards, they were, like, just something, well, kind of dirt spurting around.
Right, yeah.
And then I asked her, I was just like, why is that? I mean, do they not have developed nervous systems? I didn't realize that they were born and, like, they had, like, basic neurological function, but
Yeah.
then, like, they grow into it, basically. So I think it's interesting to see the differences, but also, like, the similarities, which what Leanna was talking about earlier with translational research. So we can now look at, like, their basic body structure and, like, their organs and stuff, and then we can try to see, like, oh, if we create mutations in this, then it'll lead to this.
Mhm.
And we can kind of see, like, maybe this kind of thing that, like, the function in a human. So.
Yeah.
Yeah.
Yeah, and she did mention that there's like limitations, like the heart only has like 2 sections instead of
Right.
the human's 4, but it's really cool to see like that in a very small model that can be like repeated a lot over a small amount of time.
Yeah, and, like, when we were looking at the differences between, like, say, like, a rat or a monkey, it's kind of like, I think it's nice that zebrafish have such fast breeding cycles, and they're similar enough to us so that it's just, like, really easy, and you can just, like, contain them and create a mutation and exactly pinpoint what kind of, like, phenotype is linked
Right.
to, like, a mutation.
And you
can
don't have to
create
wait
it.
months either. It's in the span of 5-6 days. That's really cool.
And hearing about the monkeys in research kind of freaked me out.
Yeah.
Yes,
Ooh.
she was telling stories and all the precautions they have to take. Because I asked a question about what diseases they have with zebrafish, but they have a really intense quarantine process. But with species like monkeys, you can't really, wait 3 generations.
Yeah.
So it's really scary working with them and hearing about all the different diseases. you can
Yeah.
get.
Hepatitis.
Yeah.
Yeah, the thing with the larger animal models is all you ever hear are the bad stories, but
Mhm.
it's understood that they're models for a reason. So.
Right.
Mhm.
Right. Hmm. Also, I didn't realize how secure they have to be with their facilities because they said they don't have currently a lot of problems. But I hadn't thought about like activists like coming, doing that. I had never considered that, but like I guess it's a
yeah.
Like,
problem.
we can't take pictures because PETA might be able to locate them, and, like,
Yeah.
That's
terrifying.
crazy.
Yeah.
Yeah. And it's something definitely to consider is because we're all interested in research that would probably involve animals, and I think that's something to consider is how comfortable are you using them in research. So but there are a lot of precautions that they take to make sure that they're only using the least amount of animals that they need
Yeah.
to and things
Right,
like that.
right.
Yeah, Dr. Susan Farmer was very forward about making sure the animals are taken care of, and she also told us about how she adopted one of the dogs, which was a heartwarming story.
Yeah.
That's so sweet. I
Mhm.
know.
Yeah.
And, like, they keep such good care of their animals, and they're,
Right.
like, they know that if they can, like, if they have a chance to replace them, reduce the numbers, then they'll do whatever they can to make sure that
Mhm.
they can just use it for their specific purpose
Right.
and not really, like, waste a life, I guess, which
Yeah.
I thought was really
Really
sweet.
important, yeah. And I think I wonder if people who are against using animals in research, if they understood all the measures that are gone to make sure that they're used properly, if they would be so angry about it.
Yeah, and she was saying, Susan Farmer was saying that, like, when people kind of ask her about, like, if she's okay with using animals for research, and, like, she would say, go to children's hospitals, see all those
Yeah.
children who are diseased. They don't even know that they're diseased, so they're happy, but their parents are struggling.
Right.
Like, oh, it just makes
Yeah,
me
it's
so
really
sad.
sad. Yeah.
Yeah.
So.
I mean, I'm glad that we have the chance to kind of look at something that could be used for something that's a lot bigger than just us. We can help
Yeah.
other people.
Definitely.
I think that's the
Really
most
exciting.
exciting.
Yeah. So I think that's it.
Yeah.
Yeah. So we're excited to continue on with this class.
Yeah.
Yeah. All right.
Oh,
See you next
bye.
week.